And we're live.
Great. Welcome back, everyone, to Elasmo Week. I'm your host for the week, Dr. David Schiffman. And Elasmo Week is an online outreach event about the science of elasmobranchs, sharks, skates, and rays, and the scientists who study them. This week is all about diversity. The goal is to highlight the diversity of scientists, the many ways of doing elasmobranch research, and to show off as many weird and wonderful species as possible. Elasmo Week wants to make a platform where real scientists can share their love and work with the public. And tonight's speaker will be Lauren Simonitis, who is a PhD candidate at Texas A&M University in Galveston in Dr. Christopher Marshall's Ecomorphology and Comparative Physiology Lab. You can follow her on Twitter and Instagram at OceanExploren, EXPLAUREN. For her undergraduate degree, Lauren attended the University of Miami, where she received degrees in marine science and biology. She's originally from Miami, Florida, and grew up in and on the ocean. So marine biology was a natural career path. More specifically, she's an aquatic sensory biologist interested in how animals use their sensory systems to understand their environment, interact with each other, find food, and defend against predators underwater. For her dissertation, she's focusing on the shark olfactory system, their sense of smell. And today, Lauren is going to walk you through how sharks use their senses to navigate the underwater environment. Take it away, Lauren.
Yeah, thanks so much, David. All right, let me share my screen. Okay. So hi, everybody. Thank you guys so much for coming out tonight and listening to me talk to you about the weird and wonderful world of shark sensory biology. I'm going to take you through a tour through the shark sensory systems. You can find me, like David said, on Instagram or on Twitter at OceanExploren. And because I really want to capitalize on the only good pun I've ever come up with my whole life for my name, this is going to be Explorin Shark Senses. And like he said, I am at Texas A&M University at Galveston. So before we get started, I want to talk about why this is important to me. So when I was an undergrad, I was constantly, they were constantly telling us about adaptations to life in water. So how we live differently terrestrially compared to how aquatic organisms function in the water. And I found this really interesting. I was also really interested in neuroscience. So I kind of meshed these together into this aquatic sensory biology field that I wanted to look at. So there are some challenges to studying life in aquatic systems as a terrestrial animal. And one of the biggest things is thinking about the differences in senses. But luckily, both air that we live in and water that they live in are both fluids. So we're still inhabiting fluids, they're just a little bit different. But there are some differences on how the things we see or hear or smell travel and look and smell underwater. So if we think about vision, we think about the attenuation of light. So what that means is basically how light is going to travel through water. And if you look at this picture, so it's pretty much blue. This I took at about 70 feet underwater. And you can see there's a sea turtle there. There's this vibrant coral reef, but it looks almost entirely blue. We get maybe some greens, maybe some tints of yellow if we're lucky, but we're losing those shorter wavelengths of light. So our reds and our oranges aren't penetrating as deeply. So visually, animals only have access to this end of the spectrum of light, unless they're providing their own light somehow, or they're in shallower areas. So as far as hearing goes, sound travels really differently underwater. It's really loud. So if you've ever been in an above ground pool or your head has been submerged in a tub and somebody knocks on the side, it sounds a lot louder. And this is due to how sound propagates underwater. When we think about touch and we think about ourselves, we think about reaching out, touching things and sensing things in that way. And while there are plenty of animals that participate in active touch underwater and are reaching out and grabbing things, when we think about sharks and about fish in general, we think more about them sensing the world around them, sensing the fluid around them versus actually reaching out, touching things, grabbing things. And then when we think about smell and taste, there are differences in how chemicals diffuse underwater. So to get started with our first, first I'm going to talk about two more senses. We have electroreception, which is something that is really great underwater because water conducts electricity amazingly. So animals are able to develop this sense to detect weak electrical fields. We also have magnetoreception, which is not unique to aquatic systems, but we definitely see it in sharks. And that's basically being able to sense the magnetic fields of the earth. So I'm going to go through our 5 senses plus these extra ones in this talk. Now for real, to get started, I'm going to talk to you about the shark eye morphology. And this is a generalized morphology that is also kind of seen in a lot of other fishes and a lot of other marine animals. So one of the biggest things is that they have a hard lens. So if you think about how our lenses in our eyes work, they're squishy and they change shape in order to focus light. But in an aquatic environment, we have a lot of pressure, especially for our deep diving animals. So a squishy lens is not really conducive to that. So instead, they have a hard lens. And since they can't change the shape, instead what they do is they have muscles that will move the lens further from and closer to the retina in order to focus their eyes. This also is pretty interesting because when they first started studying shark vision, when scientists first started looking at this, they would restrain sharks and sharks would pull their eyes into their head because they were trying to protect them. And when they did this, they were making themselves nearsighted. So if you look at really early literature on shark vision, you'll see that a lot of people say that they're nearsighted, but that's not true. They're emmetropic, just like us, so they can focus their eyes to see things at different distances. The other cool thing that sharks have is the nictitating membrane. So this is that third eyelid that will come up over their eye. It protects them from sediment getting in the eye. If they come in contact with something, it'll come up and protect their eye. This is a great way as well. If you're ever like if you've ever seen people working up a shark outside of water, one way that you can tell sharks that have this, if they're okay, you just spritz them in the eye with some water. And if that nictitating membrane comes back, that's a good response that the sharks are doing okay during whatever fieldwork procedure you're working on them. Not all sharks have this, but some sharks do have a secondary adaptation to this, like our great whites, which if you've ever seen in the really dramatic nature specials, when they breach and they try to bite something, they will roll their eyes into their back of their heads like you see here. Another animal that does this are the whale sharks. And whale sharks recently, this was just like a couple months ago, this paper came out that showed us that there are actually dermal denticles that are on the eyes of whale sharks. And that is crazy to me that they have these. The dermal denticles is basically what is on the skin of sharks, and they are covering their eye as well. So they're really focused on protecting their eye. Additionally, sharks do have dynamic irises, and this is something that sets them apart from bony fish because most bony fish cannot open and close their pupils like sharks can. So something that is really big in sharks of all sensory systems, all different parts of the shark sensory system is diversity, because we have such a diverse swath of sharks. We have sharks that live in the deep sea. We have sharks that live in coastal regions. We have sharks that live in the open ocean. We have sharks that go into rivers. So because there is such a diversity in lifestyle and what they're perceiving, what kind of environment they need to use their eyes to look at, there is a huge diversity of eye shapes, of pupil shape, of lens shape, of a bunch of different things. So looking at these different eye shapes and eye diversities can kind of give us some information about form and function. So when looking at relative eye size, this has been correlated both to habitat and prey style. So Lisney and Collin in 2007 looked at all these different eye shapes and correlated eye size to where they lived. So the largest eyes were in the oceanic species, primarily this big eye thresher, which actually has one of the biggest eyes of any vertebrate. It's like the blue whale and then some sport fish and these guys. So all of these big oceanic species, they're living in the open ocean where light can penetrate really easily. They have a good access to light and they're really using these visual cues. Additionally, we saw really large eyes in these benthopelagic, so these deep sea fish. So all these guys have these big eyes because there's so little light available in the deep sea that they need to catch whatever light is available to them. They really need to capitalize on it. So they have these giant eyes in order to take advantage of that. On the contrary, the smaller eyes were on our coastal sharks, such as our sand tiger shark here and our bull shark. So these guys had smaller eyes. And this is also, if we think about it, it's a little bit more due to the fact that they have so many things going on in their coastal environments. They have access to their visual cues, but they also have access to smells and to their electrical sense and to hearing. There's a lot more cues coming in, so it's a little bit more balanced. There's also a correlation between prey species. So sharks that were feeding on fish or squid or really active animals that they needed to hunt, they had larger eyes overall. So if we're not just looking at eyes, we also want to look further back, so into the brain. So Dr. Yopak looked at this in 2015, and she looked at the optic tectum and the tegmentum. So this optic tectum is this red part of the brain right here, and this is the part of the brain associated with the optics. And then you have the tegmentum, which is pretty multimodal, but is also associated with that. And overall, they saw that this was really large in those oceanic sharks, but also in those benthopelagic reef sharks and the benthopelagic coastal sharks. So that includes those two sharks with small eyes. So although they have small eyes, they do have large areas of the brain that they are using in order to interpret these signals. The smallest tecta were found in these benthic sharks, specifically the deep sea sharks. So the deep sea sharks, they have these really big eyes, but their brain that is devoted to it is smaller because they're not really getting as much of these signals. They're getting very limited light. If anything, it's bioluminescence. And then the smallest of all was this blind shark here. They're not really blind, but they do spend a significant amount of time outside of the water. So they have a really, really thick nictitating membrane that will come over their eye and protect it from drying out. So yeah. So I wanted to talk about what we know about visual behavior. So I really want to talk about Dr. Jeannie Clark. She is foundational to shark science. She is foundational to women in shark science and just women in science in general. She did so much, and I highly suggest reading books about her, such as Shark Lady, because they are fascinating. But she showed us that sharks can learn visual cues. And since then, we've learned that sharks can also be trained to learn vision-based tasks better than some bony fish. So what Dr. Clark did is she had adult lemon sharks. She taught them to just locate a square white target. So whenever they found the target, they got a little food item. So this was an example of them learning that, okay, I can associate this shape with food. Then she had them discriminate between different shapes. So like, for example, a square, a triangle, or different patterns on the shapes. So she had them just showing that they can learn more and more different and special cues. And then since then, this has really been a big area, both in research and in shark husbandry. So I know the aquarium that I work with, they visually train their sharks to respond to cues, to visual cues. So they'll put a visual cue in the water, and then the sharks will come over and get fed. That way that they're associating getting fed with a visual cue instead of a human in the water or a human hand feeding them. But yeah, this really laid some great groundwork for shark eyes and how they use them in order to learn behaviors. So there is so much more about shark vision, but I don't really have time to go over it. But there is an amazing 2018 review by Dr. Sean Collin that I highly recommend if you're interested in this kind of stuff. So moving on, we have shark ears, and they are very hard to see. So this picture always makes me laugh whenever I see it. But really, I always see that they're the third hole from the nose. So if you start at the very tip, the first hole here is the nostril or the nares. So that's the opening to the nose. And then right here, you have the second hole, which is the eye, which we just talked about. And finally, we have the ear, which is used for hearing and balance. So just like us, they use their ears to both hear and to process auditory stimulation, but as well to deal with their balance. One of the biggest challenges to learning about sharks and their hearing capabilities is the fact that sharks don't make sound. So if we have an animal that makes sound and communicates with sound, we know that, okay, they're producing sound in this decibel range, they're producing sound at this frequency, so we know what they should be able to hear. With sharks, we don't really know this to begin with, so scientists were kind of starting from ground zero. So this is basically the anatomy of the shark ear. So this is a Port Jackson shark. And just like us, they have semicircular canals. So inside of these semicircular canals, there is fluid. And this is what is responsible for angular acceleration. So when you are sitting in a rolly or a spinny chair and you spin around really fast and then you stop spinning, the fact that you still feel like the world is moving, even though you're not, is due to these semicircular canals, because there is fluid in them and they are still spinning even though you have stopped spinning. Additionally, they have the saccule, the lagina, and the utricle, which are in the center part. And this is where the center part here is where we have linear acceleration, as well as responding to sound. So if you look inside of here, you're going to see this matrix of otosonia and then an otolithic membrane beneath it, and then these little hair cells underneath. So what is happening here is whenever the shark is moving forward, so they're on this gelatinous otolithic membrane. So when the shark moves forward, the otosonia go backwards, and then that moves the gelatinous membrane, which then stimulates the hair cells and tells them, hey, we're moving forward. This is also the part that responds to the particle part of sound. So it vibrates and tells them that there is sound as well. There's also some other sensory parts of the ears, but they're not as well studied. So I'm not going to get into them right now, but these are the two main parts. But something really interesting, which is why I brought up otosonia, is in some sharks, they are actually integrating sand grains. So when you look inside of the shark ear, there are external sand grains that they're inputting into their ear in order to make it more dense and to have more otosonia to make it more sensitive. And in the Port Jackson shark, this paper in 2011 actually found that all of their otosonia were exogenous, which means that none of their otosonia were produced by the shark itself. They were all from external sources, which is crazy. But there are two different parts of sound, which also presents an issue for sharks, because the particle motion, like I said, that's the part that can excite the otosonia. But then the pressure component of sound, that's where it gets a little dicey, because most fish will hear that pressure part of sound through their swim bladder. So their swim bladder, in case you're not familiar with a swim bladder, is right here. This is the swim bladder in a bony fish. So this is what inflates and deflates to keep them buoyant in the water column and to control where they fall in the water column. But sharks don't have this. They take care of their buoyancy through a lot of different mechanisms. But these bony fish, a lot of them have what we call an otolithic connection. So that is a connection between their ear and between their swim bladder, which allows them to feel this pressure part of sound. So sharks don't have that. So we're like, well, are they even responding to that part of the sound? And so far, sharks have been shown to be sensitive to the particle motion part of sound. And this is happening at low frequencies. They are also thought to be less sensitive than bony fish in, I think, all pretty much auditory evoked potentials or physiological trials or behavioral trials. They have been shown to be less sensitive than bony fish. And we think that it's a lot due to this lack of a swim bladder. Also, if you look at the otosonia that they are making themselves, they're not as hard as bony fish, they're more gelatinous. There is evidence that sharks are able to localize sound. So experiments have shown that if you play sounds 180 degrees apart, sharks can orient to each sound. A paper in the 60s also showed that a lemon shark has like a 10-degree amount of separation between sounds. So it can localize to sounds within 10 degrees. One really cool study that looked at directional hearing was in 2007. They had a shark and they had him strapped basically into this tank. And they had shakers that could provide sound in an X, in a Y, and in a Z. So you have your X, your Y, and then the Z is underneath. So that is up and down. And they could produce 7 different directional sounds. And what they did is that they measured the auditory evoked potentials. So that is what we see right here. So here is an electrode. And basically what that means is that when the ears and the auditory system is hearing a sound, it sends an electrical signal to the brain that's like, hey, we're hearing something, we need to respond, we need to do something. So they can pick up on this electrical signal and they can get an output saying, hey, the shark is hearing something. And in this study, they found that no matter which of those 7 directions they sent a sound from, the shark had a similar auditory evoked potential. So also in this, they tested a goldfish and the goldfish did better than the sharks. So it's a little embarrassing. This is one of my favorite studies. I found this like last night while I was preparing for this talk, and I cannot believe I haven't heard this before. I think this is very cute. I think it is also just amazing that we can still have science that is really, really fun while still learning stuff about sharks. But in this, they taught sharks to recognize jazz. So they had these Port Jackson sharks, which are these cuties over here, and they had them in the start box. And then they taught them that, okay, we're gonna play jazz from this speaker here, and we are gonna give you a food reward every time we play jazz. So they taught sharks, you hear jazz, you go, you get this food reward. So then they did trials where they played jazz and they saw if the shark still swam to that food reward, and they were able to. So they got 5 out of their 8 sharks to do this. Unfortunately, the second part of their experiment, which was could sharks tell the difference between classical music and jazz, it did not work out. They could not. It was basically just up to chance whether or not they figured it out. But I think that would have been very fun, but we still learned something pretty cool. And I think this is just a great example of fun science. So going over from hearing, I'm gonna go into shark feeling. And I don't use the sense of touch for the same reasons as before, because these guys, they're not reaching out, they're not touching, they're not grabbing, they're not thinking, they're not touching in the same way that we think of. So they're more feeling what is around them. And this is due to the lateral line system. And I'm gonna give a quick shout out to my friend, my colleague, Aubrey Jones. She has some great SciComm on the lateral line system. She's in Dr. Jackie Webb's lab. It is a mecca for lateral line systems. And I am just a peruser of this sensory system. So if this is cool and you wanna know more, please check them out on Twitter. But yeah, so this is the lateral line system. And the lateral line system is made up of neuromasts. And these neuromasts are gonna sit in these canals. So here is a canal. This is running right underneath the shark surface. So here is the shark's little dermal denticles. And then underneath here is the lateral line canal. And then each of these little green humps is a neuromast organ. So what's happening here is water is coming in through these pores and it is exciting these neuromast organs. So what happens in these neuromast organs is there is this cupula on top, and it's basically like a jelly mold is how I normally think of it. So whenever the water is moving past it, that moves the jelly mold. And then underneath you have these hair cells with these kinocilium and sterocelia, and then those move. And when those move, they send an action potential down through the hair cell, which then sends it through a series of nerves to the brain that communicates the message that there is movement happening. There's water movement happening around us. So sharks have 3 different versions of this. They have superficial neuromasts, which are just these neuromasts sitting in grooves between their scales, between their dermal denticles, open to the open environment. And then for an example of where they are located in lots of different places on lots of different sharks. They're very unique to each species, but here it is on a scalloped hammerhead, and all of these little dots, each of those is a superficial neuromast organ. So this is the arrangement on a scalloped hammerhead. It is different throughout sharks and elasmobranchs in general. Next, there are pored canals, and this is our more like traditional neuromast situation. So this is like what we saw here, where there is a pore open to the open environment, water is flowing in and going through this canal, which is exciting neuromast organs. Then we have non-pored canals, and these are canals that are not open to the external environment. So it would be like this, but if these were closed, there were no gaps where water is flowing through. And this is where we get more to like what I normally as a terrestrial human think about touch. So if something is touching the shark, if a conspecific runs into a shark, if they are interacting with a prey species, if they're hitting substrate or coral reefs or something, this is what is being stimulated. So it's whenever they're being touched on their skin and there is mechanosensory pressure happening. This has less recorded functions than bony fish. So for bony fish, the lateral line system is used in lots of behaviors for lots of functions. And I wouldn't rule it out in sharks, but this just hasn't really been recorded as thoroughly in sharks yet. But there are still some great functions like orienting into a water mass or using it to find prey species. And one of the best examples of this was by Dr. Jane Gardner in 2007. And this was a really cool experiment. I talk about it a lot, but it is really neat how this is a great example of how sharks are using multiple sensory systems to come together and to find their prey. So what was happening here was they had these two bricks up here and they were sending in, let me move my little Zoom screen because it is covering the graphic. But one of these is sending in water, seawater. The other one is sending in an odor cue. So what happens is when these are piped in here, it'll create this eddy, this big odor plume. And then this allows the sharks to navigate through this eddy plume and use what we call eddy chemotaxis. So that is their ability to go through this giant odor plume and find the source of the odor. So whenever they had all their faculties together, so whenever they were able to use their lateral line system, to use their sense of smell and to use their vision, no problem. They were able to navigate through that plume and find the source. But when they ablated the lateral line system, so they basically knocked out their lateral line system, didn't give them the ability to use it anymore. They took a lot longer to find the source of the plume. And then when they took away their visual system, so when they did this in darkness, it took a lot longer. They just weren't as good at it. They couldn't really discriminate between the plume and the source. It was a mess. So it just really shows how beautifully these systems are integrated in order to form this really amazing hunting machine. So the next sense is shark taste, and that's not really what shark tongues look like, but it was honestly just a funny picture. And shark tongues aren't that exciting right now. We know that they have organs that resemble taste buds for the moment. We know that they have that. We know that they are not just on their tongue. So on these dogfish, on the spiny dogfish, they have it all around their mouths. And we know that they are able to use their mouths to discriminate between food and non-food. So there has been experiments where they cover a food item in something gross or distasteful, and the shark will put it in their mouth and then let it go out of their mouth. So we know that they're able to discriminate using their taste, but we really don't know that much. There has been some histology on these different taste buds, but this is still an area that really needs some solid research. There are other chemical senses, such as the solitary chemosensory cells. So this is a basal or primitive, if you want to think of it that way, condition where they have these chemosensory cells that are between their dermal denticles and are open and sense chemicals in the water. They also have what is called the common chemical sense. And these are just free nerve endings that are open to the water. And this is more associated with distasteful toxins or irritants. So in some studies where they tested this, they ablated the olfactory nerves. So they basically cut off access to the olfactory system, and sharks were still able to respond to chemicals. But for all of these senses, this is still a pretty wide area of research. But now we're going to go into what I am biased for, but it is the shark olfactory system, or shark noses. So this is my area of study. I'm not really going to talk about my research too much today because it is in the publication mobile right now. So I'm not going to talk about it quite yet. But I'm going to take you through general, what is shark olfaction? What do we know? Some really cool studies that I know of. So this is a schematic that I made basically because my old like outreach presentation graphic was pretty gnarly. It was like a chopped open head from a shark that a fisherman had donated to me. So I was like, let's make it a little bit more public friendly because not everybody wants to see that. So this is basically, we have here the nares or the nostrils, and this is where water is going to flow into the olfactory rosettes or the olfactory sac. They're called some different things. So in here, there are lots of different foldings and lamellae, which are covered in this olfactory epithelium. So the olfactory epithelium is covered in olfactory receptors. And these guys are basically receptors that sit and they wait for water to come in with a chemical. And when it comes in, it binds together and it sends a signal out and says, hey, I'm smelling something. So it creates an action potential, which sends out an electrical signal to the brain. So these are happening here. And then that signal travels up through the olfactory bulb, through the olfactory tract, and into the brain. And that's basically an outline of how this works. So this is a bonnet head shark. And as Jada said in her shark, it is the best, her talk, it is the best hammerhead shark. So I appreciated that shout out, but there is a huge wide diversity. So as you can see, these guys have pretty big noses. But then we get to our great hammerhead that has an even bigger nose. And we have this giant olfactory sac, these huge olfactory tracts that come to the brain. And then we compare this to our spiny dogfish. They just kind of have these little olfactory sacs. They're kind of more short and stout. And then a little olfactory bulb here, which is this little olfactory tract. So there is a wide diversity, just as we have seen in all of our sensory systems, a pretty wide diversity of what this morphology looks like. So what the shape looks like. So Dr. Kaijura did a study on basically comparing the olfactory capabilities of hammerheads with these really wide, long noses compared to non-hammerheads with these short, little pointy heads, which give them a shorter nose. And they found some things like the nostrils being farther apart helps them to differentiate between odor gradients. So if odor is coming from one side, if your nostrils are farther apart, you can tell the difference a little bit better. They also found a prenarial groove in hammerhead sharks, which allows for water to come in at a higher volume and provides for greater flow. They also found more olfactory lamellae, so more folding in that olfactory epithelium, but they did not find more olfactory epithelial surface area. So there wasn't really a difference in the sensory part of this structure. And then furthermore, Dr. Tricia Meredith and Dr. Kaijura did do electro-olfactograms where they looked at all of these different elasmobranchs and they used an EOG, so an electro-olfactogram. And basically what this does is it's kind of similar to those auditory evoked potentials we were just talking about. But these guys have electrodes that go right outside of the nostril. And again, like I said, whenever the nostril smells something, it sends an electrical signal up to the brain. So those are a bunch of action potentials happening across that nose. So if you put an electrode in salt water right outside of that nose, you can get kind of a summation of all those action potentials happening. So you can tell like, oh, the shark is smelling something. And overall, they found that there wasn't really a difference. Even though there was a difference in morphology, there wasn't really a difference in sensitivity. Also, they found that they're not that much more or less sensitive than bony fish. So sharks kind of have this reputation for being these like swimming noses, and they're just controlled by their sense of smell. And they, it's like in Finding Nemo where he smells blood and he just can't control himself, and they can smell a drop of blood in like the whole ocean. And they're really sensitive. That is true. They do have these super powered noses, but so do bony fish. So they're not that special, even though I still think they're cool, but physiologically, not that special. But the other part we can look at is the olfactory bulb morphology. So we've looked at the outer parts of the nose, and now we're looking at the bulb. So that is the parts here in red. And again, this is Dr. Kara Yopak. Same thing as when we looked at the optic tectum and the tegmentum, but now we're gonna be looking at the olfactory bulb. And again, there were some trends with lifestyle. So the largest olfactory bulbs were in these coastal oceanic, like this tiger shark, and in these deep sea sharks. So if you think about these deep sea sharks, they're not really getting a lot of visual information. So they're really relying on this olfactory system. So a large percentage of their brain is devoted to this. It reflects that. The smallest were in these reef-associated sharks, like this white-tipped reef shark, because again, these are the guys that are getting all of these different signals. So if you think about it, their brain is a little bit more evenly distributed. They don't have giant olfactory bulbs. They don't have giant of one or the other. They kind of have a more evenly distributed brain because they're getting all of these different signals. They're getting light, they're getting sound, they're getting electric, they're getting lateral line. So their brains are a little bit more balanced. Also, it was pretty small in these oceanic sharks, like these purely oceanic sharks, like the big eye thresher. So we've gone through our typical 5 senses of vision, of hearing, of touch, of smell, and of taste. So now we're going to talk about the 6th sense and the 7th sense as well, but I did not have a Bruce Willis movie for that one. So first is electroreception, and I feel like this is always on the nature specials what is marketed as the 6th sense, and it's this amazing adaptation, which it really is. It's really cool. So these are ampullae of Lorenzini. You can see them here and here as well. So this is one of my bonnet heads. So another one that was donated by my fishermen. Thank you, community partners, for helping me do science. And all of these little pores and grooves and canals, these are all the ampullae of Lorenzini. And if you were to open them up and look inside of them, if you've ever done a shark dissection, a shark head dissection in school, you'll see that they are filled with jelly. And this jelly is some of the most conductive, naturally occurring substance on Earth. It is super conductive. It's not quite as good as what we've made, but as far as naturally occurring stuff, it's pretty great. This allows them to both detect biological and oceanographic electrical signals. So biological electrical signals are when you move a muscle, sends off some electrical signals. When your heart beats, it sends off electrical signals. Even the imbalance of ions between inside of your body and the outside environment, that is an electrical signal. So they're able to detect these really weak biological signals. Additionally, they can pick up on oceanographic electrical signals. So water is a polar molecule, which means that one side it's positive, the other side is negative. And whenever ocean fronts pass each other, they create this little charge. And I always picture it like this scene in Finding Nemo, where you see like the EAC moving through. And I kind of think that that's what they see through their electrical oceanographic senses. So this is a really, really classic experiment that shows how they're utilizing this sense. And I'm going to take you through this figure piece by piece. So if we start at the top, here we have our shark. And here we have a flounder, and this flounder is buried in the sand, but maybe they can still see the outline. Maybe they can kind of suss it out. So they have access to their visual senses, they have access to their chemical senses, and they have access to their electrical senses. So the shark is able to locate it really easily, gets it good. So then here we have the shark once again, but now we've put this little casing around it, which is stopping it from seeing the fish, any outline, any sort of movement of sand. So we've blocked it from view, but there is still water flowing through, which gives a chemical signal. And he can still see through with his electrical sense and pick up on the electrical signals of the fish. Now here we are with an electrical blocking and basically a sealing little contraption around it. And in this, there is no electrical signal, there is no chemical signal, there's definitely no visual signal. So the shark just kind of swims by. But then finally here, this is just the electrical signal. So there is no more flounder. There is just these little electrodes here that are mimicking the biological signal of the flounder. And the shark is still going and attacking it. So here is a pretty cool experiment, and I love this video. I always show it to my students when I teach, and I think it's a great example of how they're using this electrical sense to hunt. So first, I'm gonna just explain what's happening, and then we'll watch the video. So this is another study done by Dr. Kaijura, and here we have a carcharodonid shark, so a pointy-headed shark, and then we have our hammerhead shark, so the long hammer, and we're gonna see how they're gonna go around and respond to activated electrodes in the sand that are mimicking these biological signals. So first we're gonna see it from the side, and then we're gonna see it from overhead. So here we have our little carcharodonid, and there it is trying to attack this electrical signal. So then from the front, we see him swimming, and then the electrical signal is turned on. He's going around and around, trying to bite and eat the electrical signal. So here with the hammerhead, something else I want you to notice is how, look at these nice tight turns. So the other day, if you listen to Jasmine's talk, she was talking about the head and what it is used for, this hammerhead. And you can see that the hammerhead here is really helping them make these really tight turns. So there he is making these super, super tight turns around this electrical signal. Yeah. So moving on to our last sense of the night, this is magnetoreception. And like I said, this is not something that is exclusive only to aquatic organisms. Lots of terrestrial organisms have this as well. And this is the ability to sense the Earth's magnetic field. This is used in both navigation and homing. And we think that it's based on this magnetite-based magnetoreceptors in the visual system and in the brain. So they first found these in rainbow trout, actually. And we do have behavioral evidence that this is found in sharks as well, that sharks are able to orient to the magnetic field of the Earth and are able to respond to it. So in this study here, they trained the sharks to respond to an artificial change in the magnetic signature of where they were. So basically they had them in a tank, and then they knew that, okay, this is the normal magnetic signal. And then they artificially changed it. And whenever they did that, they would reward the sharks with food. So they would teach them, here, you get food at this point. So whenever they would do that, the sharks would be looking for food. However, then they put these giant magnets on some of the sharks that blocked their ability to sense the Earth's magnetic field. And when this happened, they were unable to sense that shift in the artificial shift in the magnetic field. And with that, we have gone through a pretty quick, pretty down and dirty tour through these shark sensory systems. I do want to just reference every study I've talked about and some things that have helped me. So if you're watching this, pause, find something you found interesting and read a paper, because they are amazing. And I just want to end this by saying, please support minorities in shark science. If you're watching this either live or on YouTube afterwards, you're using their YouTube channel. They are an amazing organization. They are so very important because a lot of times shark science feels very male and very white. So it's very nice to have this community, and you can find them on Instagram or on Twitter, and you can go to their webpage and you can find the donate page that you can monetarily give if you are able to. And with that, I just want to say thank you guys so much for joining me and taking the time out of your Wednesday night. And yeah, thanks.
Thanks, Lauren. That was great. So just as a reminder for everyone that on Sunday evening, we're going to have a live Q&A with all of our presenters for Elasmo Week. There will be more Elasmo Week talks tomorrow evening, same shark time, same shark channel. But Sunday night, all the speakers will be all together for a conversation and Q&A session. So if you have any questions for Lauren or for any of the other speakers, leave them on this YouTube video that you're watching, or tweet them at Lauren or at Elasmo Week. Thank you very much for watching, and thanks for